Okay, so I'll continue today with the rest of lecture 5, and then we'll get into lecture 6. As I mentioned, I would really like us to be able to get through lecture 6 today, or at least for half the class on Thursday, so we can briefly go through some of the exam material that you should really focus on. And so last time we stopped about, we actually stopped the last class where we were talking about telomeres, right? And how important telomeres are for maintaining the ends of eukaryotic chromosomes, right? Because it's not circular, so because of the way DNA replication works, you could lose ends of your chromosome every time the DNA replicates, and that's where telomeres are important for maintaining the integrity of the ends of those chromosomes. So there's other, I would say, repetitive sequences that are normally present within organism genomes. And one of an important type of repetitive element or repetitive sequence is referred to as transposons. And transposons are unique in the sense that they're able to move from one location in your DNA to another location in your DNA. Okay, so it can move from one part of your chromosome to another part of your chromosome. And for that reason, they were referred to as mobile elements, right? They're mobile, so they can move. They were originally discovered in the 1940s by Barbara McClintock, who was actually a Mays geneticist working at Cold Spring Harbor in New York. And she was studying chromosome breakage. And that's how she was able to discover these unique elements that were present within the maize genome. And since that time, and she actually won the Nobel Prize for this discovery because it's a huge discovery, meaning that every organism that we've studied since then contains some aspect of transposable elements, maybe a different variety or a different group, but it has been found in almost every organism that has been studied so far. In bacteria, these transposons are referred to as insertion sequences, right? So bacterial transposons are referred to as insertion sequences. And they're, quite frankly speaking, the most simplest form of transposable elements. They contain only the elements necessary for their own transposition. And those elements that are necessary for transposition is the short inverted repeats at the ends of the transposon, right? So that's part of the transposon, these inverted repeats. And they contain at least 2 genes coding for an enzyme that helps them to move around in the genome, right? So and that enzyme is referred to as a transposase, and it's responsible for cleaving the DNA and reinserting somewhere else in a new location. The way that this transposition works is that once the transposon moves to a new location within the DNA, it will cut that DNA sequence. And by cutting that, the way that it cuts the DNA sequence creates duplication at the ends of that flanking insertion. So the new DNA, that's the new transposable element that inserts in that new DNA location, the ends of those contain a duplicated sequence, okay? And one copy of the sequence, again, flanks the insertion on each side after the transposition event. And it's really kind of markers to know that the transposition event has occurred. And just to describe this in a little bit more detail, so you have a bacterial plasmid. This is the DNA sequence that's present on that target, right? This target gets cleaved, this DNA sequence. Because of the way it gets cleaved, it's creating these staggered cuts similar to what you see when you use certain types of endonucleases that creates the overhand, right? So it's cleaving and creating these overhands as well. The transposon inserts at this particular location from wherever else it's coming from. And once this insertion happens, these gaps need to be filled in, right? And once these gaps are filled in, what you have is these sequences repeated on the same ends. These are they're direct repeats because they read the same sequence in the same orientation, right? So ATA It's also ATA here. So direction is the same, so they're referred to as direct repeats. So initially, when transposons were discovered, they were referred to as selfish DNA because they were considered to be replicating at the expense of their host and not provide anything useful to the host. With bacteria, we know now that they can provide useful information, such as valuable genes, antibiotic resistant genes in bacteria, and confer a new type of resistance to bacteria that wasn't there before. So we know that bacteria plasmid requires, you need to have different types of antibiotic resistance genes. Some of these can be engineered, but if you're for cloning purposes, but in general, they can acquire them by the use of these transposable elements. So imagine that you have a donor plasmid that have kanamycin resistant. You have a transposable element, which is called TN3. And this transposable element contains an antibiotic resistant gene, ampicillin. This transposable element can then insert into a target plasmid. So here is the target plasmid containing another antibiotic resistance gene, tetracycline. And it can insert into this new plasmid and provide now this ampicillin resistance gene. So you now have a plasmid that's acquired a new antibiotic resistance, making it more usable and more versatile in bacteria. So there can provide some useful benefit to the bacterial host. And the way that transposons in general are able to move around in the genome has been well studied. So there are different types of transposition mechanisms, the way that they move or transpose. They are usually referred to also as jumping genes, right? So they can move from one location to another, and they do not always have to do this by completely removing itself from that location, okay? So it doesn't necessarily have to move from one place and go to another place. But when it does that, it's usually referred to as a non-replicative transposition. So it's cutting, moving to a new location and pasting in that new location. So cut and paste, okay? So both strands of the original DNA move together from one place to another without replicating. There's another type of transposition which involves DNA replication, and that is referred to where one copy remains at the original site and then a new copy, which has been replicated, moves to a new site. And that mechanism is referred to as replicative transposition or copy and paste, right? So you're making a copy of that transposable element and that copy goes and insert into a new location, compared to where the transposon just cut from one location to another and move there. In replicative transpose, the other cut and paste is very straightforward. So it's just cutting and cleaving and going into a new location. And in replicative transposon, so this is an example of a transposon in bacteria, so TN3, which you saw previously. Again, contained about 2 genes that's required for its transposition. So these are the transposis genes. It also contains a recombination site that's important for it recombining and remaining in the new target location. And it also contains an antibiotic resistance gene. So this BLA is very similar to ampicillin resistance. It's just another name for ampicillin resistance gene. So you have your target DNA, you have your transposons, your 2 plasmids, right? What usually happens is that these 2 plasmids will fuse together, right? And form this single plasmid, or what we refer to as a co-integrate, right? So you can see that this is that part, and then this other part is here, right? So they kind of fuse together. This co-integrate then gets resolved by recombination. And that resolving this by this recombination events then copy the transposon onto that target. And while the original part of the plasmid also retains a copy. Okay, so it's replicating it. So it's going from, you know, the original plasmid to the target location. And at the end of that event, you get back that original plasmid containing the transposon, and then one copy is gone into the target gene. Okay, so this is kind of how the this replicative method works in bacteria. And the other way that you can have this moving around of these transposable elements, in the sense that if you have a non-replicative transposition, as in the sense of this other transposable element TN10, the idea is very similar, except it's being cleaved from the original location to the new location. So you start the same way. You have two plasmids, one containing the black one, the blue containing the transposable element. The green is the target location, the target plasmid. They will fuse together like you saw previously. However, the way that they get resolved requires cleaving of the DNA, right, on both strands of that original plasmid. So this new plasmid pretty much contains that transposable elements. It really removes it from that donor plasmid. And when this gets resolved or separated, you now have the transposon in the target plasmid. The original plasmid or donor plasmid has a gap. That gap gets repaired and the transposon is no longer there. And now you have it in the new location. And this is really the kind of the idea behind non-replicated transposon, and particularly in bacteria. The mechanism, of course, is a little bit more complicated in eukaryotes in a way, but the idea is very similar. And in understanding how these transposable elements are able to move around in multicellular organisms or eukaryotic genomes, I mentioned that Barbara McClintock was the geneticist that actually discovered these transposable elements in maize. And the way that she was able to very easily discover that there is something moving around in the genome that's creating a phenotype was looking at kernel color. Okay, so you guys all are familiar with maize. You know that maize can have many different color kernels, and a lot of the variation that you see in the kernels for these corn are due to transposable elements. Okay, and the way that this works is that you have, this is the chromosome, you have the gene that codes for a color kernel, okay? And this gene is a gene that produces anthocyanin. Anthocyanin is usually found in many berries, right? So like blueberries or raspberry, right? It really provides a pigment. And this anthocyanin gene is present in maize, which normally gives a purple color to the maize kernel. Because there's a transposon residing right next to this color kernel, by random chance, if this transposon moves and inserts into that gene, it creates a mutation, right? So the gene becomes a mutant. It's no longer functional. It cannot produce that purple color. So you have a white kernel, right? And that's what you see here, okay? This is due to a transposition event, okay? And the reason you have so many different colors is because it's not happening in every cell at the same time, right? It's a somatic insertion. So it's not happening in every one of these cells at the same time. So not all kernels will have either purple or white. In the event that this transposable element then moves again, so it comes out of the gene that's coding for the purple color, you can get restoration of that purple color. And these are what you're seeing with these spots. And you see the spots here, right? Where you see that purple spots suggest that the transposition has happened again and the element has removed from this gene. So you'll have, in some of the cells, the color will be restored and you have these spots, okay? What's special about this transposable element is that it cannot move by itself. You guys recall when I talked about the bacterial transposable element, we said that it requires two main features, right? It needs the genes or the enzymes for its transposition, and it also have these inverted repeats at the end of the transposon. This element, which is called DS for dissociation, because it's really dissociating the color, is dissociating from its location, inserting into this gene and disrupting the color, requires an activator element, another transposable element. This other transposable element actually is providing the transposes for this gene to move. So in the absence of this transposable element, this gene, this transposable element DS is not able to move. Okay, so they work together, and this is well studied in organisms where these transposons are present. Okay, and it's a very classic transposon pair that's used for mutating many genes, and it's still being used widely in plant genetics today. Okay, so the AC is the element that's providing that transposes, so it's referred to as an autonomous element. So it can provide a transposes for DS to move. It can also move itself on its own, okay, because it has all of the information that's required. And this is what you see here in an actual image of a corn kernel, right? Again, you have the mutation happening, and you will have the mutation happening, you have white kernels, right? So this is where you have all the white, the DS is inserted, you have white kernels. Where you see the small spots, right? This is where the DS element has removed from this gene, the C gene. So you have these spots. It happened late in development when the kernel is already kind of developed and most of the cells have already divided. So that's why the spots are kind of smaller. Larger spots suggest that they've happened earlier in development. So more cells have that element removed or excised from that gene. So it's much larger. And here's where you have complete restoration, because again, it's really happened really early in development in the germline. So you're having the complete restoration of that purple color. This type of transposon moving into genes that's coding for important colors within plants or other types of phenotypes, like visible phenotypes you can see. You can also see this in plants such as snapdragons, right? Which is Anthorhinum, where you have, sometimes you see these very striped or variegated patterns within the flowers. And this is really due to transposable elements moving around in the genome of that plant that's giving this variation in flora color. In eukaryotes, you have different types and classes of transposable elements. Plants and humans have very similar classes of transposable elements. They vary depending on the sequence to some extent, and that's what really distinguishes them between the species. But in terms of the class, they're very similar. So plants have DNA transposons, which are referred to as class 2 elements, right? They're DNA transposon because they move with a DNA mediated transposition. They insert and move to another location, okay? So almost all DNA transposition, all of DNA transposon move by a cut and paste mechanism, okay? So they insert into the DNA and they can be removed from the DNA by cutting itself out and inserting into a new location. And these are also present in humans as well. Similarly, you have what I mentioned, AC, like the AC element, which they have the transposes that's required for its transposition, and also the inverted repeats, which is usually drawn kind of inverted. So these arrows are pointing out towards the DNA. So these are inverted repeats that's present on the transposon. So this entire region is a transposon. The arrows that's pointing in the same direction here and here are the direct repeats. These are not part of the transposon. They're just what happens when the transposon inserts in the DNA. Okay, and that's very important for you to know the distinction between those two. And this transposon have all the information required for its transposition, so it's called an autonomous element. Non-autonomous elements have the inverted repeats and maybe some of the transposes, but not the functional transposes. Okay, so it's not able to move from one location to another without the help of another element that contain that transposes. This is also found in mammalian organisms, in other eukaryotic organisms, particularly mammals. The other major class of transposable elements are class 1 transposons. They're present in both plants and animals. They have, instead of inverted repeats, they have long terminal repeats on that transposon, and also the enzymes required for their transposition. Okay, you can also have autonomous elements or non-autonomous elements where it doesn't have the enzymes required for its transposition and relies on these autonomous elements. These are also present again in human genomes as well. These type of class 1 elements. There's another type of element that's present in both plant and animals, which are called non-LTR elements. So they don't have these long terminal repeats. So you see it's absent here, but they also have the genes that's required for its transposition. These elements, transposable elements, vary in abundance or the copy number within the genome. So within the human genome, 21% of the human genome contain these type of transposable elements. And which is a huge portion, right? So you guys recall when I talk about how the human genome is very large in terms of the sequence, right? It's like 3,000 megabase pairs, but you also have only about 20 to 30,000 genes. A lot of that other regions within the genome are filled with transposable elements. They're not obviously actively moving around in our genome because you could imagine we're all probably be dead, right? Because they will be inserting like crazy into important genes that's required, for cell division or other aspects. So when transposons start becoming mobile or becoming active for whatever reason, they can cause mutations, right? So a lot of mutations, whether it's related to cancer or other types of mutations, that we have studied can be due, and in some cases are due to transposable element moving and inserting into an essential or important gene. So they're not as we thought in the beginning. So in the beginning, we thought they were just junk DNA, right? They're just these pieces of DNA that's in our genome that came from somewhere, right? We don't really know where it came from. And this just happened and they're not doing anything to the host. But now we know that they're very important because they regulate many aspects of many different mechanisms in our genome, okay? And they could be detrimental, okay? So most times we have transposable elements there. And someone asks, what is the purpose of having so many transposable elements? The purpose, some of them are important because maybe they have important elements within their sequence that helps in some way. But for the most part, they're there because it costs the cell way too much energy to remove them. And if they start moving around in our genome, the cell can quickly shut them down, right? So transposable elements are not commonly active. Our genome have ways to keep them from moving around, right? So it's really tightly regulated in our genome, but they're there and they just increased over evolutionary time for different reasons, okay? So the two main classes of eukaryotic transposable elements are these class 1 and class 2, very similar to bacterial, replicative, non-replicative type of transposition and transposons, as we mentioned. So class 1 elements are the elements which move more by replicating, so they make a copy of themselves, right? So they first make an RNA intermediate, And this RNA intermediate is what gets reverse transcribed by reverse transcriptase, which is encoded in the transposable element, right? So it's one of the enzymes that's contained in the transposable element. And it makes a DNA, and that DNA copy is what gets inserted into the new location. So in a way, it's copy and paste like what you saw before, right? Because it's here, it's just making a copy, an RNA intermediate. That RNA intermediate gets reverse transcribed, and that's what's inserted into the new location. DNA type transposons or class 2 transposable elements, these actually remove from the location, right? So they excise from their location and they go and insert into a new location. So you kind of just cutting and pasting, okay? And this is what's found in both plant and animal genomes, and is very common in many other eukaryotic genomes that we've looked at. One hypothesis is that these transposable elements maybe, it's like a chicken and egg question actually, but one hypothesis is that these transposable elements at some point might have evolved or or got integrated from viruses, right? We get infected by viruses all the time, by retroviruses. And when you look at the structure of many different types of retroviruses, what you find is that they have these transposable elements within their genome, long terminal repeats, different enzymes within that transposons that's required for their transposition. So this is a retrovirus. And what you see is that when you look at transposable elements in either yeast or even Drosophila or in humans, you find similarity to those genes, right, within those genomes. So what's different is that between the transposable elements and a retrovirus, it does not contain this envelope gene. The envelope gene is the gene that makes it infectious, right? So transposable element lack this envelope gene. So whether or not they lost it or viruses co-opted it, we don't really know, okay? So that's kind of what one of the main differences between the two types of transposable elements compared to retroviruses. So those are transposable, again, they're very special within our genome, in many, many genomes, and they've been well studied. We have found a way in plants and also in other systems to use it to our advantage to study gene function, because we know what the transposons are, we know how they move, so we use them to create mutations to study, a gene that we're interested in. So we've leveraged all of the mechanism, the type of transposition mechanisms to our advantage for many different aspects of gene function. So just kind of summarize what we've talked about in lecture 5, right? We talked about types of DNA damage, which you want to understand the different types of DNA damage, right? How do certain types of radiation and chemicals cause mutation? Understanding that a mutation is different from DNA damage, right? And what that difference is. How is DNA damage repaired, right? The types of repair mechanisms will vary depending on the damage. What is the molecular mechanisms of crossover, right? So timing is important, right? Depending where in the cell cycle will determine the type of repair mechanism for double-stranded break repair, right? So you can either have non-homologous end joining or homologous recombination. Understanding the importance of telomeres, not just what telomeres are or what telomerase is, right? But understanding why it's important for us to have these telomeres at the end of our chromosome is important. And then with transposons, what are transposons? The mechanism of transposition in bacteria, and also the mechanism of transposition in eukaryotes, and how they help to spread antibiotic resistance in bacteria, okay? Some questions that you want to think about in terms of the exam when you're studying for the exam, right? I feel like this should be a help to at least guide you to the type of questions you could see. What are some of the common examples of DNA damage? You guys probably don't remember this right away, and some of you do, so just very quickly, just so we can get through the lecture, what are some of the common examples of DNA damage? Just give me 2 examples of DNA damage. UV radiation, Okay, and thymidine dimers. So what does UV radiation do? So you'll know this by the exam, right? You need to know what, it disrupts the backbone. Okay, and how does it do that? Okay. It break the hydrogen bond, knocks it out. Okay, so there's many different answers, but so I really want you guys to think about how, how UV radiation might alter the DNA, right? So UV radiation in general forms exactly pyrimidine dimers, right? Other types of alkylaging agent or gamma radiation will cause a different type of DNA damage. So you really need to know the difference between those different damages by the different chemicals, environmental chemicals. What are the mechanisms that repair damaged DNA? What are just two of the mechanisms, just two mechanisms that will repair DNA damage or damaged nucleotides? Recombination will repair double-stranded breaks, okay? And non-homologous end joining, correct? But that will repair double-strand breaks. But what about the other types of DNA damage, right? What about alkylation or pyrimidine dimers? How might you repair those type of DNA damage? Yeah, so using some kind of excision repair, right? Or some kind of, base excision repair or nucleotide excision repair. And understanding how those mechanisms work, not necessarily the particular enzymes, but the type of reaction that's happening. You already told me the two different mechanisms of double-strand breaks, which you mentioned. What are the ends of chromosomes, right? Know what they are. Knowing the enzyme that's used to extend these chromosome ends, right? And that's very related. And what is telomerase composed of? You guys should remember what telomerase is composed of, because that is actually important for its function, right? To be able to extend the DNA. So thinking about what's in the composition of telomerase is important. You will definitely see that type of question. And why is this type of mechanism important for cell division and maintenance, right? You need to really understand why it's important, not just that it's at sequences to the end of chromosomes. Okay. What are the two we just talked about? DNA transposon. So what does DNA transposon contain? There's two things that are important. Yeah, inverted sequences, or inverted repeats. Yeah, so it has the inverted repeat, and what else does it need? What else is part of that transposon?
The enzyme, but I forgot its name.
Yeah, the transposase, right? So you need to know that enzyme, right? The transposase, right? Two genes for transposase. You need to know those enzymes. So that's the composition of the transposons. And what happens to the flanking sequences when the DNA inserts? They duplicate, okay? And because they duplicate, they're called direct repeats, okay? And they're flanking that new DNA sequence, okay? So that is kind of where we stop for all of lecture 5. And I'll try to see if I can do another polling. I don't know if it's going to work because I have not figured out all the details of this yet. So let's do another question about, again, I'll do this 1 minute, so just so we can continue. So this question is really based on DNA synthesis and replication, which you should all be thinking about and you should all remember because you've already been through this in your discussion session. Okay. And I'll end it when a minute has gone by. Okay. All right, so yeah, so the majority of you have polymerase, and definitely polymerase is what's required, right? Is the enzyme that's responsible for DNA synthesis during during replication. Okay. In terms of DNA damage, let's do this, and then we can do the last one while you guys are not thinking about the other question, which we just talked about. What are the two types in which double-stranded breaks are repaired? Okay, so we're kind of like a mix between the two. So yeah, so the two ways in which double-stranded breaks are repaired is non-homologous end joining and homologous recombination. Okay, base excision repair. So maybe nucleotide excision repair can, well, that yeah, both of those will very rarely create double-strand breaks, right? I mean, you usually remove the strand or the nucleotide that's kind of damaged for DNA damage. So for double-strand breaks where both strands of DNA are broken, you want to repair it by non-homologous end joining or homologous recombination. Again, that is highly dependent on when it happens during cell division. Okay, will determine which mechanism might be more preferred. Okay. Let's remove it. Okay. So again, try really going through the material and trying to understand what is happening for each of these aspects of either DNA replication, the enzymes that are involved, and also the same with DNA damage and DNA repair. So I'll continue on now. So we'll switch gears now, right? So we've been talking about DNA replication, DNA damage, what can happen when if a base gets modified by a chemical or if a random break in the DNA. And about also transposable elements, which can create mutations depending on if they're inserted into a gene, as you saw with the kernel color, right? Where it creates a mutation in the color kernel gene, the color gene, which you no longer have that purple kernel. So we talked a lot about DNA replication, repair, genetic recombination. The next step in the flow of genetic information is now going from DNA to RNA, okay? And that process is what we will talk about for the next couple of lectures. We'll talk with bacteria first and then in eukaryotes. But this is synthesizing RNA or transcribing the information from the DNA into RNA, okay? Before that gets made into a protein. Besides the fact that this happens in both eukaryotes and also prokaryotes, which is bacteria, the way that it happens vary, and you'll see that as we go through the different steps, being that the RNA that's made in eukaryotes are subjected to a range of additional processing steps that you don't see in bacteria, okay? So in a way, bacteria is a little bit simpler in terms of transcription. And the important parts of transcription in any system, right, is the enzyme that promotes the transcription or that's important for transcription. And that enzyme is RNA polymerase, right, similar to DNA polymerase, but for replication or DNA synthesis. You need other elements that are important for transcription, and you need to start the transcription, make the RNA transcript or extend the RNA transcript for all of the information that's encoded in that DNA. And then you need to stop because you don't want to keep making RNA for all of the DNA that doesn't even code for anything, right? So you need to be able to stop at some point when that information is no longer important. Okay. So this is just an overview of a gene structure, and this is very universal to any gene structure, right? So what you have is a region within the DNA that's referred to as the promoter. This region signals the start of the gene for RNA polymerase to recognize and bind to, okay? It gets recruited to this location once everything that's required for transcription is present here on this DNA and this promoter region. Then you start to transcribe the RNA. And that's referred to where, and that tells you where does that RNA start, is referred to as a transcription start site. And that will get transcribed all the way to the end where it's a transcription stop site or termination site. This is the end of what the information you need to make, okay? Within this region, you have a coding sequence. The coding sequence, again, it's an open reading frame, starts with ATG, ends with a stop codon. This is the information that in the end will get translated into a protein, okay? So this all get transcribed, but only this information that's in this coding region or this ORF is what gets translated into a protein. In addition to what's present on that DNA, you also have regions in the promoter that's important for helping to regulate transcription. Okay, so it's binding site for positive or negative regulators of transcription, right? So they either promote transcription or reduce the level of transcription of this particular region or gene. So let's begin. Polymerase is absolutely the most important enzyme because without polymerase, you can't transcribe anything. It's very simple, of course, in bacteria because there's only one RNA polymerase. So RNA polymerase use DNA as a template, right? Because you're transcribing that DNA sequence into RNA. And it catalyzes the formation of these phosphodiester bonds that links the nucleotide together to form a linear molecule or RNA, okay? The growing RNA chain is extended one nucleotide at a time in the 5' to 3' direction. This is important, right? Because you're making a single-stranded molecule of RNA, and it could only, RNA polymerase can only extend 5' to 3', okay? Because it can only extend DNA 5' to 3' direction, you cannot transcribe both strands of DNA. This is not replication where you can do leading and lagging strand. You can only transcribe a single strand of that double-stranded DNA, okay? That's important, so just keep that in mind and we'll get back to that later on, either in this lecture or the next lecture. In bacteria, there's only one polymerase. There's multiple polymerases, 4 to 5, depending on the organism in eukaryotes, okay? And also because you have different organelles in eukaryotes, the mitochondrial polymerase is different from the polymerase that transcribe nuclear genes, okay? So different organelles. So with everything, right, these are a lot of experiments that were done back in the day to understand, you know, what are the polymerases that are present in bacteria, how many are they, what do they look like, right? And to do that experiment, what was done by a chemistry group was they looked to see what is the composition of RNA polymerase in bacteria. So this is a gel, right? It's a polyacrylamide gel, very similar to the gel that was used to look at the Sanger sequencing reactions, right? So it's a very similar composition, was acrylamide that was used. What's also, this is a gel which we refer to as a protein gel. It's a protein gel because the bands that you're seeing here are all protein bands, right? This is not DNA, this is proteins. And this gel is also ran using a electricity, right? So negative to positive. Proteins, the charge of the proteins are made to be negative by the use of this detergent called SDS. So protein gels quite often are referred to as SDS gels. And that's because you use this detergent to add a negative charge on the protein and also to linearize the protein so they can run through this gel. And because they're negatively charged and linear, they can be they can migrate to this positive end and you can separate the proteins. And that's the way they separate the RNA polymerase structure. And they found that this RNA polymerase actually have multiple subunits, right? So it has two beta subunits. One is beta prime and beta. It also have a sigma subunit. This is a non-specific band. That's why there's an asterisk here. It also have an alpha subunit, and it also have an omega subunit, which is relatively small, which you can't really see on this gel, but it exists. And these are just different purification methods of that RNA polymerase, of the bacterial RNA polymerase substrate. So you have different bands, but these are really, lane 1 is really what you should focus on. Lane 5 is where they just purified only this sigma subunits. That's why you only have a single band here, okay? But this is just to show you that you have multiple subunits that's present within the RNA polymerase, and this was actually experimentally discovered. And again, this is 1969, very early on, and it tells you the different sizes. Proteins are usually, the units for proteins are usually kilodaltons or daltons, but more quite often kilodaltons are usually the units for these, for proteins, if you're going to be looking at the protein abundance on a gel, okay? And in equivalent, if you can think 1,000 base pairs of DNA, probably have, because every base, you have 3 base pair per codon or amino acid, right? So you could have about 333-ish amino acids. And that's usually equivalent to 37 kilodaltons, right? And that's how the conversion is made based on the size of the protein. And so based on that, we know that that RNA polymerase is similar to DNA polymerase in the fact that it's a multi-protein complex. This complex contains 2 major parts. So the catalytic activities in the core, so alpha, there's 2 molecules of this alpha subunit, and beta prime, okay? So this makes up the core, okay? The holoenzyme, which is everything that's required for full function of this polymerase, includes this sigma factor, which is important for promoter recognition, right? You need to recognize the promoter so you know where that polymerase is going. And then there's also this omega subunit, which the function is really unknown. So we're not gonna see this, we're not gonna talk about this for the remainder of the lecture. And the core also contains helicase activity, as you'll see, because you need to kind of somehow unwind the double-stranded DNA to be able to start synthesizing that RNA, okay? So this polymerase multi-protein complex really do have these 2 separate functions. The sigma factor is absolutely required because it really enhances the binding to the DNA. Otherwise, the polymerase just really, just very weakly bind to the DNA, okay? And it's not able to synthesize or transcribe the RNA efficiently. The way that they determine that this sigma factor is important for interacting with the DNA and transcribing the DNA, right, is by doing experiments, biochemical experiment, isolating the sigma factor from the rest of that multi-protein complex and looking to see whether or not it can transcribe efficiently DNA, okay? So the core enzyme is the enzyme without the sigma subunit, was not really able to transcribe. So we were looking at transcription activity here. 0.5 is pretty low. So the core enzyme doesn't contain the sigma factor. You have a very low value, suggesting that it's not able to efficiently transcribe that viral DNA. However, if you have the holoenzyme, which contains the core and the other protein complex, you can see that you're able to transcribe it. And this is really a control. They use calf thymus, which is a nicked or a nicked DNA, which is a control here because with nicked DNA, they're more susceptible to being transcribed. It's non-specific, random transcription. And even in that case, you can see that the core enzyme is, it can transcribe this core enzyme, but very similar to what you see. A little bit better with the core only, but you also is very similar to what you see when you have the whole enzyme together. Okay, so it works equally well on both types of DNA, whether it's nicked or the intact or unnicked DNA, which is the T4 here. Okay, so this is how they were able to discover that this core subunit is important for transcribing intact, fully functional RNA to transcribe it. And it needs to be also part of this whole enzyme to really fully function. What we think that the sigma factor is doing based on a three-dimensional structure analysis is that this sigma factor, so this is the whole enzyme, this is the RNA polymerase without the sigma factor, right? And you can see the subunits are kind of a little bit separated and not really forming a complete structure. With the addition, which this doesn't really show you where the sigma factor is, but with the addition of the sigma factor, this whole complex is able to come together and form kind of a loop that the DNA can actually be part of. Okay, so it kind of surrounds that DNA and forms a loop attaching it to the DNA. And this is kind of a schematic of what that really is telling you, that the sigma factor really helps to seal the polymerase around that DNA. So double-stranded DNA, you see the sigma factors are in contact with this double-stranded DNA, and the rest of that polymerase is attached as well, right? So it really helps to stabilize and wrap around the DNA. So how could you test if this sigma factor helps the polymerase stay attached to the DNA, right? So do you guys know of, are you guys aware of any experiments you can do to figure something like this out? What would you need to be able to do to determine whether or not this sigma factor helps the polymerase to stay attached to the DNA? So polymerase is a protein, a multi-protein complex. You want to see whether this multi-protein complex attaches to the DNA. Any idea? You could do a gene knockout, which you can, of the sigma factor, right? Yeah, you can do an isolated sigma factor. So you can do both of those, right? You can do gene knockout, you can remove the sigma factor, right? And then study the function and see, is it important or not, right? So there's an experiment, experiment that you can do kind of in the test tube. All the suggestions you guys are giving are great because that's really what you can do in the living organism, right? And those experiments have actually been done. In the test tube, very easily, we can do an experiment which we refer to as a filter binding assay. Filter binding assay or nitrocellulose binding assay, because nitrocellulose is actually the filter, the name of the filter that we're using, really helps you to look at, is this protein interacting with DNA, right? So you're doing protein DNA interaction, okay? And the way that this experiment works is very simple. It's very simple components. You guys already mentioned you isolate what you need and then you test it, right? So double-stranded DNA, if you add double-stranded DNA, solution of double-stranded DNA to a filter, it will go right through that filter into your test tube, okay? So if the DNA is labeled, you can check the filter to see, is the DNA on the filter or not, right? And if it's not on the filter, that means it flowed through into your test tube, okay? So if proteins are large, proteins cannot go through this filter. So if you were to pour your protein solution on this filter, it gets stuck on this filter, okay? And doesn't pass through into the flow through. However, the proteins are not labeled, so you can't really tell whether or not the proteins are on this filter or not, okay? However, if you did a protein and DNA complex, so you mix your DNA with the protein, in this case, you would have your sigma factor, your mutated form of your sigma factor, and you will have your DNA, right? So you mix it together in a test tube. Your DNA is labeled, your protein is not, you mix it together, and you pour it through the filter. What do you expect to happen? Well, you have scenario one where you have the flow through here, or you would have this one where it's not flow through. Yeah, exactly right. It gets caught and it stays on the filter because the protein is bound to the DNA. The protein cannot go through the filter, and that means the DNA cannot go through the filter either, right? Because it's bound to that protein. So it gets captured in that filter. And then you can check the filter and you will find radioactivity. This is a classic experiment that's usually done to check whether or not your protein can bind to your DNA. If it cannot bind to your DNA, the DNA is just going to flow through, right? So it's very specific. It's not just random, right? You're going to just use random protein and random DNA. It will not work, right? So it's really a way to determine whether or not your protein binds directly to your DNA. And that's exactly what they did with the sigma factor. So this is the result of that particular experiment I just discussed, right? So you have labeled DNA, and it's telling you the percentage that's retained on that filter. So the higher, 100%, that means most of your DNA was bound to your protein. So you have really high levels of radioactivity, okay? If you don't have, if the DNA didn't bind to the protein and it flows through, then you have very low level of radioactivity. And that's exactly the experiment that was done with the sigma factors. And the holoenzyme, right? Remember, the holoenzyme is the enzyme that contains everything, right? So you incubate your proteins with your DNA, and the holoenzyme with the DNA, you can see high levels of radioactivity, very, very stable interaction. So your protein and the DNA is bound, and you have high levels of radioactivity. If it's only the core, there's no sigma factor, right? You mix it together, that binding goes completely down, and you can see with time, right? So you have the DNA, you collect the sample as soon as you put the solution on, and that's why this is high. But then you wait some time and you see that everything is flowing through, right? So you're losing all of your DNA, you're getting less radioactivity, right? So it's not binding. And that's because the core is not there, right? So the sigma factor is not there to help to bind to DNA. So they concluded that the sigma factor helps RNA polymerase complex to stick to that promoter regions in that DNA, okay? And these usually, these two examples just showing you the half-life of those proteins. And that's really, when people talk about half-life, is how long does it take for half of your activity to be gone, okay? And 30 to 60 hours is a very long time. The holoenzyme is highly stable. You have a very less than a minute half-life. That means the core is very unstable, okay? It's not as stable, okay? So that's the experiment that they did. And in addition to say, okay, this sigma factor is really important for recognizing that promoter region and helping the polymerase to bind or attach to that DNA, they also want to know whether or not this was temperature dependent, right? Because we know that the activity of at least DNA polymerase is related to the activity, is related to temperature. So they also want to test the same type of analysis to see, is the RNA polymerase binding to the DNA in fact temperature dependent or independent? So they did a very similar experiment as what we did, the filter binding assay, but after exposure to different temperatures, right? So you did 37 degrees Celsius, 25 degrees Celsius, and 15 degrees Celsius. Which of these would you expect to have stronger binding? So what do you expect? So 37, okay, so there's 37 and there's lower temperatures. So both are important. So at 37 degrees Celsius, right? You will have higher temperature will promote the DNA melting, right? So the DNA is going to separate, and that way the RNA polymerase is going to be able to bind better, right? So as a separated DNA. Lower temperature, the double strands don't melt as efficiently, so they're a little bit more stuck together. So you have less RNA polymerase binding to the DNA. Okay, so higher temperature, you get more binding or encourage RNA polymerase binding, and lower temperature you have less. And that's why you see this labeled DNA going down, right? So as the temperature is lower here at 15 degrees Celsius, you get a decrease in binding, so you don't have as much radioactivity, right? You have some radioactivity, but then as you pour that flow through, more and more of that DNA is going to flow through as the temperature is lower, while at 37 room temperature, over time, it's very stable, the binding to those tubes. This is temperature dependent reaction. So the way that RNA polymerase recognizes the promoter and binds to it, together the core and the sigma factor forming this whole enzyme kind of scans along the promoter, right? Looking for something, or the DNA looking for a promoter, searching for the promoter, where to bind. You'll see later on it will recognize certain sequences in the promoter. Once it recognizes certain sequences, and this is really promoted by this sigma factor, it really strongly binds to the DNA. Okay, so you see the DNA going through it, it kind of wraps around binding to the DNA. Okay. In addition, once it binds to the DNA, this sigma factor helps to transition from this closed promoter complex to an open promoter complex. And it does this because this whole enzyme, as you guys recall, have some kind of helicase activity. So it's able to unwind the DNA and melt the DNA at the promoter, forming this open format. Okay. How does RNA polymerase know where to bind? As I mentioned, it's scanning along the promoter. It's looking for certain sequences. We know what these sequences are because we've studied many promoters in genes, and we know what types of sequence are present where the RNA polymerase is bound. Okay, so there's core promoter elements that are important for binding, for this polymerase to recognize and bind to. Okay, this region is common to all bacterial promoters. It's 6 to 7 base pairs centered around about 10 base pairs upstream of the start of transcription. So transcription starts here. It's 10 base pairs upstream, so it's referred to as -10. Okay, so this -10 box, it's usually 6 to 7 base pairs. Here we're showing 6, highly AT rich, right? And it's bound here. Also, in addition, there's another sequence that's exactly -35 base pairs from the transcription start site. That is also important for recognition of that promoter. So eukaryotic core promoters, -10, -35 of these sequences. And this is present in thousands of bacterial promoters that we have checked. And we also know that this spacing is absolutely critical, just because of the way that the polymerase binds and the size of the polymerase enzyme. And so the spacing is important. And by comparing many thousands of promoters, these sequences that are present here, when you compare them across many different promoters, you can create what we refer to as consensus sequences. If you ever hear anyone talking about consensus sequence, a consensus sequence is determined by comparing multiple similar sequences, okay? And then you look to see how often each nucleotide is present at a particular location, okay? And that's how you generate the consensus sequence. It is often shown like this in the scientific literature. In DNA sequences, if someone writes it in a gene structure, in this case, it's also written in this context. Sometimes it's have uppercase or lowercase, right? The way it's written is really depending on this consensus sequence. So this is the consensus sequence. If you notice at the -10, you have T and A, you have these lower T, A, and A, and then you have a higher in terms of the height. The height determined percentage, okay? So T and A is almost always present at this first and second position. These three positions can vary, and that's why it's like lowercase here and lowercase here, and then A and T here. So this can also vary, and this T is almost always present at this location, okay? So that's how consensus sequences are determined based on how often you find, if you look at 20,000 bacteria promoters and you look at this position, how often do you see a T relative to the other bases, right? And it's really based on percentage. In addition to these two regions on the promoter, right, the -10, -35 box, there's additional elements present in the promoter that's important for regulating transcription as well. So the E. coli promoter that encodes this RNB gene, which you'll see a lot later, is just a gene in bacteria. And in that promoter, you have this up element, which is upstream of this -35, and it's usually -40 to -60 upstream of this transcription start site as well. And it helps to stimulate transcription by 30 times, like 30x. Like, so it really increases transcription significantly. And it is part of the promoter because it's recognized by RNA polymerase, okay? Besides the up element, there's additional cis elements, and cis elements are really just elements that's present on the same strand of DNA and the same location or chromosome of DNA, okay? So usually they refer to as cis element because they're present on the same DNA molecule. And you have additional cis elements -60 to -50, which you don't see here, so it's like somewhere upstream here, that are called enhancers. These are not bound by RNA polymerase, but they're bound by transcriptional activators, and we'll talk about those also later on. And they're also important for helping to enhance transcription. Yes, exactly. Yeah. So the up and basically it stimulates transcription. It doesn't necessarily make it go faster, but it really stimulates it and increase how much RNA is being made. Yeah. When you look at the sigma factor, which we know is important for binding to the promoter elements, the -10 and the -35 box, again, this is looking at all, like multiple sigma factors, I should say, not all, multiple sigma factors in different bacteria. We've sequenced, we've cloned them, we've looked at the sequence, and you can see that there have similarities to some extent, right? So you see that this domain 2, I'm calling them, or region 2, is highly conserved, is present in many different bacterial species. You also have this blue that's present in many different bacterial species. The one that we mostly focus on in E. coli is sigma factor 70. And this sigma factor 70 is the primary sigma factor used in E. coli. These sigma factor, even though it's part of the RNA polymerase, the way it recognizes regions within that promoter is based on those regions in that sigma factor. So you have region 1, 2, 3, and 4, which recognizes the -10, 4 would recognize the -35. And these are the regions that are present here, right? And those are the regions that are recognizing the DNA sequence in that promoter, okay? Region 1 prevents the sigma factor from binding by itself to DNA, right? So the sigma factor just can't bind by itself to DNA unless you force it in a test tube, but in general in the cell, it needs to be part of the entire RNA polymerase, the whole enzyme to be able to do that. And then region 3 is involved in both the core and the DNA binding, right? So the rest of the whole enzyme needs to be in contact here as well. How that specific binding happens, right? So you have the sequence-specific recognition is achieved. These regions in that sigma factor, so 4.2 or 2.2, for example, recognizes actual nucleotides within these regions. It's highly specific, right? So these arginines are recognizing G and C, and threonine and glutamine is recognizing this T at this -10 position, right? So it's highly specific of what the recognition is for this to happen. Otherwise, you will just randomly be able to transcribe anything, right? So it's really highly regulated and specific. Right? How are the other promoter elements recognized by RNA polymerase, right? So we know that the sigma factor recognizes the core promoter elements, the -10 and -35. So what recognizes the up element, right? So there must be something in the polymerase to recognize the up element. And this appears to be the alpha subunit. So there's 2 molecules of the alpha subunit that's present in that core polymerase. And that's the protein that helps to, and that's a subunit that helps to recognize that up element. And because it's part of that entire polymerase, right? So you have your sigma factor, right? Bound to your 35, -35, -10, beta, beta prime, and you have your 2 alpha subunits. They are able to bind to that up element. You notice schematically how these are, the distance is important, right? So you can see that it needs to be able to attach to this up element, which is -40 or -60 upstream, right, of the transcription start site. So that's why those positions need to be conserved, right? To be able to have this polymerase to be efficiently be able to recognize that binding. I'm going to stop here before going into the experiments that they did to get in there, and we'll continue this because we have to get through this. We're going to continue this in the next class. So I'll stop right here in terms of where the how the alpha subunit recognizes the up element. And before we take any questions, since you guys have been distracted and completely engaged about bacterial transcription, which is completely awesome, and you'll see why it's completely awesome. And I did say, yeah, the up element is -40 to -60 upstream of this transcription start site. Okay. And before we continue, I have one last poll that I wanted you guys to be thinking about the transposable elements since you've been distracted on transcription, and I'll be able to test how great your memory is, right? So here is one of the questions that you can possibly see. I'm giving you guys these practice questions, but you'll realize that some of these questions, it's quite possible you will see them again on the exam, the very same question, right? So just keep that in mind. So what happens to the flanking sequence where the newly inserted transposable element inserts? It's very mixed, interesting. Very mixed, okay. I'll try to see if we could get all 46 of you, or most close to as much as possible. Okay, it's very mixed. It's very mixed. So we talked about what is the composition of a transposon? What is the composition of a transposon? I know you guys still remember this. Right, you have the inverted repeat, yes. And what else? And the transposase. Okay, so that's the transposon. And when it inserts in the DNA, right, you cut the DNA. When the DNA gets cut, what happens to the DNA that gets cut? It's very similar to restriction endonuclease. What happens then? Right, you have sticky ends, right? You have these overhangs, right? So then your transposon goes in and you have these sticky ends that needs to be filled in. So then what happens to those ends on each side of the transposon? They get duplicated. And because they're duplicated, they're in the same orientation, right? So they're direct repeats. So the correct answer here would be where 21% of you guys got it, generates direct repeats, right? So that's exactly what happens. Okay. So try to remember this. Just for the record, my entire undergraduate and also graduate work, my PhD, was based on transposons. So I love transposable elements. So you will see several questions on transposable elements. Okay. So just so you know. So try to understand what is the composition, the mechanism, particularly eukaryotes, and we will be able to, and also how they insert in the particular location. That's super important for you to know. So yes, so the information covers on Thursday, once we get through to the end of lecture 6, will be what will be covered on the exam. I will try to have a study guide ready by Thursday for you guys. I will have office hours still on Friday. Try to do the practice questions, even if you don't know how to answer them. Do the practice question, homework questions that's available at iLearn until 11:30, because some of the questions there, you could possibly see them again. And so there will be no surprise for you in the exam. Okay, so just try to take advantage of those. I will have office hours on Friday at 3. It might be a little bit later. I will let you know for sure what time that will be if I decide to go later. And I'm also willing to extend it beyond 1 hour to an hour and a half, depending on, I'll take a poll on Thursday in class to see how many of you are able to come longer. Take advantage of your TA's office hours and also discussion session as well for this exam, because this is a lot of material, and exam 1 is usually very challenging for many people just because it's a lot of material and it's a lot of more synthesis rather than memorization, right? You really need to understand what's happening in a sequencing reaction, in a PCR, in a cloning reaction, in a vector. You really need to understand those those aspects of the material. Mm-hmm. Mm-hmm. Okay. Any questions? Again, I'll stick around for a little bit. If you guys have specific questions, you can answer.
Professor?
Yes.
So for the -35 and -10 sequences, are they similar to the ones in eukaryotes, or that?
Yeah, it's very similar to the one in eukaryotes, especially the -10. So you will also see this TA box present in eukaryotes when we look at eukaryotic transcription, and also that -35. Yeah. Right. Eukaryotes don't necessarily have an up element. They have other types of element, but it's not referred to as an up element, but there's other types of cis elements. But the -10 and -35 is present in pretty much most organisms that we've looked at.
Oh, we're going to talk about eukaryotes later, right?
Yeah, so we're going to talk about eukaryotes right after this, after bacterial transcription, yeah.
Gotcha, thank you. Mm-hmm.
Doctor, could you re-explain that last poll question? I'm still a little confused on that whole process.
About the transposon.
Yeah, like with what you said happens to the flanking, like when is the transposon then inserted?
Mm. Do you see the, or could you see this new slide, the generating host DNA direct repeats?
Uh-huh.
Okay, so you can imagine, right, that you have a plasmid or a DNA sequence, right? This is double-stranded DNA. This is a recognition site where the transposon will insert. What happens is that transposes enzyme, right, within the transposon, cuts this DNA here and here, just like how some of the endonuclease is cut, right? It cuts on the two strands in opposite sides, right? So it will cut the DNA because it wants to insert there. It cuts the DNA. Imagine that this plasmid is being now pulled apart, right? Leaving these overhangs, right? These are complementary sequence. The transposon inserts. On each side, you have these pieces of the sequences that are here. They get filled in, right? Because it's opposite strands and they're complementary. This left side gets filled, the bottom strand will get filled in, DNA polymerase will come in and add all the bases, and this gets filled in, right?
Mhm.
This other side of the transposon that's inserted, that also gets filled in. But the sequence is exactly the same because in this case, the bottom strand is being filled in from this, and here, the bottom strand here is being filled, the top strand is being filled in using this bottom strand. So it's exactly the same sequence that's duplicated on the sides. And they're the same direction, right? So they read ATA, they're going in this direction. They're reading exactly the same. And that's why they refer to direct repeats. Compared to if it's inverted repeat. If this was an inverted repeat, then this sequence would be flipped over horizontally.
Oh.
Right?
Oh,
And that's kind of, if you go here, where it shows you schematically what that looks like, right? So you have the transposon, you have the arrows usually pointing out because these are direct repeats. These are part of the transposon. The transposons are made that way. They're important for recognition of the transposase. Like for some elements which don't have a transposase, the only way the transposase can recognize that, say, this element is a transposon, or like in the case with, remember AC and DS, the only way that AC can recognize that DS is the transposon it needs to move is because of these inverted repeats. That's part of the transposon. And then you see the arrows pointing in the same direction. Those are the direct repeats.
oh.
And these direct repeats gets created when a transposon inserts. Inverted repeats are already part of the transposon.
Oh, okay, so they're going 5 to 3' in both directions.
Yes, pretty much. That's the direct repeats. Yeah.
Oh, okay. I think I was just confused on what the like flagging end meant.
Yeah.
So that little, when they're missing their complement, that's what's considered the flagging end?
Which one?
Like going back to that diagram that's where on the slide that says generating host direct.
This one?
Yeah. So like on part C, that's the flagging end.
Right. So this is the flanking end. This is the flanking. This is flanking ends. Yeah. This is a transposon, and this is the flanking ends. This transposon, right? So this is direct repeats, right? This is a direct repeat. This transposon still have inverted repeats because it's part of the transposon. You don't see it here, but it's part of the transposon.
Yeah. Okay, yeah, I understood all of that part. I just wasn't sure, like.
Yeah, this is just flank. Yeah, this is the flank.
Yeah. Okay. All right. Thank you so much.
No problem. See you Thursday.
See you Thursday.
Thank you.